I'm Bill Rapisi, President and CEO of the Lymphatic Education and Research Network, and I just want to welcome you and say how delighted we are to have people from all over the world here today for this brilliant symposium. It's our honor to be able to present these to you free of charge, but that's only possible thanks to LE&RN donors, and that's where you come in. This is the time of year when LE&RN has its annual appeal. And right now you have an opportunity to double your impact thanks to a donation from Ian Lehman and Stephanie Free, who gave us $50,000 as a match. So when you see the button to give to the appeal during the symposium, please give generously so that we can continue to bring this programming to the world and continue to fight against lymphedema, lipedema, lymphatic malformations, and all lymphatic diseases. Thanks for your time. And remember, when you see the button to double your impact, please click it. Thank you.
Thank you for joining us today. I imagine we have a diverse audience with different expertise and different background. First, let me, I'll show you a couple of slides that supposed to, show you before my presentation. And now, a few words about myself in case you don't know me. My name is Guillermo Oliver. I'm originally from Uruguay, but I have been here for a long time. And I'm currently the director of the Center for Vascular and Developmental Biology at Northwestern Feinberg Cardiovascular Research Institute in Chicago. My lab has been focusing on lymphatic biology for over 20 years. At the time that the field was still pretty much ignored, except for those impacted by lymphatic diseases. And the reason we started to work into this field was because our serendipitous identification of the first lymphatic specific marker, a transcription factor called PROX1. And that became a very valuable tool because it allowed us really to identify the lymphatic vasculature. Not only PROX1 was a valuable resource from that perspective, but turned out that inactivation of PROX1 in mice generated the first mouse model completely devoid of a lymphatic vasculature. And what that demonstrated was that PROX1 was critical for the development of the lymphatic vasculature. And during these 20 years, my lab, and as well as many other labs around the world, has been really dissecting the process of normal lymphatic development. In addition to that, what we have been doing lately is to focus in other aspects of lymphatic biology. It has to do more with the lymphatic role in normal and pathological settings, in particular in the cardiovascular field. And my goal today, and you see it on the title, is basically give you an overview of where the field is standing today. I would say in the last decade or so, our understanding of the function of the lymphatic vascular has really changed from the traditional point of view to today, say that the lymphatics are a very hot topic in which lymphatic function is critical in a variety of not suspected roles in normal and pathological settings. And what I'm going to do today, during most of my talk, is give you a brief overview of those novel functional roles of lymphatics. But then also try to tell you a little bit of what we are doing to try to see if we can better understand how symptomatic or asymptomatic lymphatic defects could also be correlated with a variety of associated comorbidity linked to these novel functional roles of lymphatics, which I have been telling you about. So let's just start then. So just again, I don't know the background of the audience, but just a very general overview. Basically, we have two major vascular systems, the blood vascular system in red, and the lymphatic vasculature in green. And both are lined by endothelial cells. That's what they have in common, among other things. And as I said before, the reason why we started to focus in this field was when we identified almost 20 years ago this gene called PROX1. And what you can see here is the dermal lymphatics of a mouse stained in purple, labeling the blood vasculature, and in green, the lymphatic vasculature labeled with this marker, this antibody called PROX1. So what about the anatomy of the lymphatic? Well, the lymphatics are mostly distributed the entire body, as you can see here in this drawing. Basically, you have lymph nodes and then larger and smaller lymphatic vessels, which you will see a little bit more in detail in this diagram here. So basically, the lymphatics that you see in panel A are connected to the blood circulation at the level of the thoracic duct. And basically, the smaller initial lymphatics are the ones that, absorb fluid and transported to larger collecting lymphatics that drain on the lymph nodes, and they eventually go back to the blood circulation. We have two major flavors of vessels or lymphatics: the smaller initial lymphatics, which are on panel B, whose cell junctions are not as tight, therefore facilitate the entrance of fluid and macromolecules through those junctions, which are called bottom-like junctions. And these initial lymphatics eventually connect to the larger collecting lymphatics, which have valves that push the fluid in a directional manner, such as I said before, they will go back to they will go through the lymph nodes. Here you see it in panel B, where they will filter the fluid and eventually go back to the circulation. Please pay attention to panel E, because I'm going to talk a little bit later about these specialized lymphatics, which you can find in the smaller intestine. And these are called lacteals. One major role of lymphatics is the absorption of lipids. And the lipids are absorbed mainly through these intestinal lymphatics called lacteals. And these lipids are packed in a smaller particle called chylomicrons. And these chylomicrons, again, will be absorbed through these lacteals and eventually will transport the lipids that will be absorbed through the blood circulation. So this is a very brief description of the anatomy of the lymphatic system. So again, the most classical and traditional view of lymphatics that we have seen in almost every textbook for forever is that basically normal conditions, basically they are required to drain lymph fluid from the extracellular spaces. Basically, they absorb lipids from the intestinal tract, maintain fluid homeostasis, and transport white blood cells and antigen-presenting cells to lymphoid organs. And in disease, mostly what we know is that if lymphatics are malfunction, you can have both types of diseases, which are the congenital acquired forms of lymphedema, particularly, or acquire, for example, after removal of lymph nodes in radiotherapy. And also they play a major role in cancer through the migration of metastatic cells through the lymph nodes. Those are the most traditional, well-known roles of lymphatics. Just one thing I forgot to mention at the beginning. If you have questions, start to write it on the chat. At the end, I will read the chat, but also we may be able to, if you can raise your hands, and I may, you can unmute yourself and ask questions in person. Anyhow, going back to what I was saying, here is the traditional roles that we are all familiar about lymphatics. And the classical one is the immunity inflammation through the transport of immune cells, and which is depicted here. That's the classical role of lymphatics, the transport of immune cell. The other major role, as I said, is in cancer. So basically metastatic cells from the primary tumor enter through to the lymph nodes, and eventually they are transported through the body to, that's another of the classical role of lymphatics that we're all well familiar with. And the other one that is also very, many of you are well familiar, which is basically in the case of lymphedema, in which is one of those genetic lymphatic disorders that is generally characterized by the unilateral swelling of the extremities, as is, you can see it here on the leg. And this is important for what I'm going to tell you later. One of the features of lymphedema, of the affected area, is not only the swelling, but this is also followed by the increase in the amount of adipose tissue around the edematous area. So that's another typical feature of lymphedema. So these, once again, are the major roles. Thanks to the work of many labs around the world, in the last 20 years or so, these are the genes which are listed here. You can barely read it, but close to 20 genes have been identified, whose, if mutated, they are responsible for different type of lymphatic malformations. And again, you can, these have helped us a lot to better understand the causes leading to these alterations, and in some cases, eventually to better understand and better treat and prevent maybe some of these disorders. So this is all what I wanted to tell you about what was known and what we all know about lymphatic function. What I want to focus most of my talk is what is in the title of this recent review I wrote with a few of my colleagues, is the novel functional roles of lymphatics. Some of them were unexpected, and the more we learn about this, the more will allow us to really prevent and treat a variety of disorders. And in this diagram, I summarize a little bit the ones I'm going to be telling you today, which are the ones we learned in the last 10 years or so. One has to do with the role of lymphatics in obesity and metabolism. The other is on irritable bowel disease, particularly Crohn's disease, in glaucoma, in cardiovascular diseases, and also in the CNS, in neurological disorders. And I will tell you briefly, but there is no doubt that there are many more novel functional roles of lymphatics that will be identified in years to come. And I will briefly tell you about one that my lab have really recently identified as well. So let's start with this role of lymphatics in metabolism and obesity. I don't need to stress how much of an impact obesity has in health, and how much obesity is a prevalent disorder in a variety of conditions. And of course, obesity the major cause of obesities are our diet and lack of exercise. And but there are in certain conditions forms of obesity for which there is not a clear cause of why people increase the amount of adipose tissue in certain conditions. And the reason we started into this is because I told you before, this gene that we discovered years ago called PROX1, when you don't have it, mice have no lymphatics and they die as embryo. However, one finding was that when you remove only one copy of PROX1, the animals survive, but eventually become extremely obese. As you can see it in this picture here, this is a heterozygous mouse. You can see it's much heavier than the control littermate. And at the time, we didn't know the reason for why these animals become obese. And the last thing we thought was about lymphatics. And these animals, if you look at them, they don't exhibit any obvious feature of lymphatic defects or lymphedema. They just are obese. So we started to characterize these animals in detail, and it was obvious that the lymphatic vasculature in these animals was affected, was not normal. You can see on the top panels, more or less, how normal lymphatics look in the skin. However, at the bottom, you can see that the lymphatics in the heterozygous animals are not normal. They don't form a nice network. They are not connected in certain areas. But not only they look different, they function differently. The function is abnormal, because normally when you inject a dye in the leg, you can see it in panel A, that's how the dye should be transported by the lymphatics in a straight line, as you can see here. However, in the rest of the panels, you can see that basically the patterning of these lymphatics is really compromised in the PROX heterozygous mice, and in some cases, the dye is not even transported. So what this told us was that indeed the lymphatics in these fat PROX heterozygous mice were really defective. So could be that this defect may be relevant for the obesity phenotype in these animals. In the past, there has been, in fact, when you start searching the literature, there is a lot of evidence of crosstalk between lymphatics and adipose tissue. In fact, it is known that, for example, lymph nodes and larger collecting lymphatic vessels are commonly surrounded by visceral fat and adipose tissue. This is a well-known fact that we all knew, but we never thought that it was a kind of crosstalk between lymphatics and fat cells. What I told you before, for you to remember, was that adipose tissue accumulation is normally observed in the edematous tissue of patients with lymphedema. It's a typical feature of lymphedema is the accumulation of fat. Again, what was the reason for that? We still don't know. It was never clear. But again, what this indicates that there is a crosstalk, a correlation between defective lymphatics and fat accumulation. Also, we know that at least in obese mice, the opposite is also true. If animals are extremely obese in certain conditions, in certain diets, impair lymphatic function, as you see it here, and lymphatics become leaky. And finally, obese patients are at higher risk of lymphedema due to reduced baseline lymphatic clearance. So bottom line, there were some evidence that suggested maybe the reason for obesity in our animals is because of this defective lymphatic function. And turns out that in our animals, when they're born, many of them, they die because these lymphatics are so defective that they break, and all these lipids, which I told you are transported by chylomicrons, start to leak out into the thoracic cavity or the abdominal cavity, and these animals eventually die. So again, supporting the proposal that lymphatics were very, very defective in these animals. And you can see it in these images, again, that there are some holes in the lymphatics in these animals. You can see it in all these images by immunostaining or by electron microscopy. Once again, supporting the idea that something is really wrong in these animals, although they don't show lymphedema. And the area where we see most of the leakage or most of the defective lymphatics is in this area I told you at the beginning, which are called lacteals in the small intestine, which are these white vessels that you can see here pointed by the arrows. And that's the ones that transport most of the lipids via the mesenteric lymphatics. And as I said before, the way these lipids are transported is in these particles called chylomicrons, and they enter through the lacteals and eventually they go back to the blood circulation. That's the way lipids are absorbed. So to test if indeed these particles were leaking out of our defective animals, Natasha Harvey in my lab at that time just fed these animals with a fluorescent tracer, removed the intestine and put it under the microscope, and where she was able to see that these chylomicrons, these lipid particles, instead of being as normal, being inside the lymphatics, they leak out to the surrounding tissue. And that was a major surprise, to be honest with you, at that time. Furthermore, where she was able to demonstrate using in vitro culture systems of pre-adipocytes, if she has these leaking chyle, these lipid particles, to pre-adipocytes in culture, that was sufficient to promote adipocyte differentiation. And that was an important finding and surprising finding at the time. And furthermore, when we started to further dissect among those lipids of those chylomicrons, what is the adipogenic factor by doing metabolomics analysis and mass spec, basically we were able to determine that free fatty acid in particular and lipids are the main factors responsible for this adipogenic feature of the chyle, particularly oleic acid, linoleic acid, palmitoleic acid, and palmic acid are abundant in lymph, and those are all capable in vitro to promote adipogenesis. So more or less, this is where we stop. But based on all this data, what we proposed, and this is almost 15 years ago, at that time, that in mice, and at least in this particular mouse model, that has no features of lymphedema, is totally asymptomatic from that point of view, we propose that leakage of chyle, of this lipid-rich particle, is responsible for promoting de novo adipogenesis, particularly in the visceral area, and that eventually promotes to these animals to become obese around 4 months of age. So that's when they start increasing weight, and that's so that's what we proposed at the time. But obviously the question at the time, okay, that's great, but does this happen also in humans? Is there something similar? And that was not an easy task, and I will tell you a little bit more about our efforts to address that in a minute. But anyhow, I want you to remember that at least in this mouse model, where we demonstrated that was defective lymphatics are responsible for fat accumulation. So this is the first of those novel functional roles that we somehow deal today, or we recognize today. And there are evidence accumulating by other labs that really there is a role of lymphatics in metabolisms and obesity. So the other functional role that also was discovered 5 or 6 years ago was the role of lymphatics in glaucoma. You can see here healthy and a glaucoma eye, which the optic nerve is dilated. And basically the reason for this defect was because there is a hybrid lymphatic vessels in the back of the eye called the Schlemm canal, which mutations in a variety of genes basically makes this vessel defective or is lacking completely. And that results in increase of ocular pressure and eventually glaucoma. And these days, there is a couple of companies that they are developing eye drops that eventually may restore vision by restoring the function of this Schlemm canal. So again, there's another unexpected or surprising novel functional role of lymphatics that we didn't know. Another one that has a huge impact lately, and there are major papers published quite often, which is in the CNS and in neurological disorders. it was rediscovered that on top of the meninges, there are lymphatics, which are called meningeal lymphatics. And basically, a number of papers have determined that these lymphatics play major functional roles in health and disease. And in particular, I summarize a few of those papers here. In health, has been shown the meningeal lymphatics are very important to drain the cerebrospinal fluid, but also that there is compelling evidence that these lymphatics may also play a role in disease, particularly in a variety of neuroinflammatory disorders, including, for example, Alzheimer's or Parkinson's disease. So this is an area of active research because, I don't need to tell you much about the importance of those diseases. But again, it was something that was totally unexpected. A few months ago, another aspect that was shown in a couple of papers was a potential role of lymphatics in hair growth, in hair regeneration, as lymphatics were shown to be attached or very close to the stem cells of the hair follicles, and they may play a role in the regrowth of hair. So again, another role of lymphatics that was not known a few years back. And whether this is also similar in other stem cell niches in other organs is not yet clear. Another one that is data keeps piling up is potential role of lymphatics in irritable bowel disease, in particular in Crohn's disease. In Crohn's disease, there is an inflammatory disease in which one of the typical features is the abnormal accumulation of fat in the distal part of the intestine, and it's called creeping fat. And a number of papers by a different lab have recently showed that indeed lymphatics in the mesenteric lymphatics in that region of the intestine are in fact defective and have holes in patients with Crohn's disease. And has been seen that basically the application of a pro-lymphangiogenic factor, VEGF-C, basically improves the condition in models of IBD in mice. So basically, there is a number of evidence suggesting and supporting the idea that there may be a role of malfunction in lymphatics in IBD, and one could even speculate about the role in this creeping fat. And finally, and I'm going to spend a little bit of time on these, another, beside obesity, which I told you at the beginning, and the number of people, obese individuals in the world, another major cause of death in the industrialized world is cardiovascular disease. And a number of evidence in the last 5-6 years have really linked the lymphatic vasculature to cardiovascular disease in normal and pathological settings. And basically, when I mention cardiovascular disease, I'm particularly talking about, for example, plaque formation in atherosclerosis or heart attack after myocardial infarction. Those are the most typical forms of cardiovascular disease that are major cause of death. And as I said, in the last few years, there have been a number of papers that really demonstrated the role of lymphatics, the unexpected role of lymphatics, if you like, into that. Basically, has been shown that cardiac lymphatics respond to injury when you have any of those conditions. For example, heart attack, lymphatics regrow, expand into the injured area of the heart. And furthermore, it has been shown that that growth of lymphatic improves cardiac recovery. Particularly has been shown that also by injecting adenoviruses with these pro-lymphangiogenic factors like VEGF-C, so you expand the number of lymphatics in the heart, basically stimulates the resolution of the inflammatory condition in the heart after the heart attack. And also improving lymphangogenesis reduce the fibrotic tissue and the edema, and improve cardiac function in a variety of animal models. So there is evidence piling up of really lymphatics play an important role in improving the recovery after heart attack. The major arguments of how they do that is that obviously, basically, they reduce the inflammatory response by bringing immune cells to the injury area. That's the classical concept that we have. However, in my lab and recent work, we tried to really further understand whether that's indeed how our lymphatics operating in the heart in these conditions. And all the cases I told you before, basically what they provide is evidence that if you have more lymphatics in the heart, that improve or facilitates the resolution of the injury, the fibrotic area, and the inflammation. What we decided to do, as I told you before with obesity, to take advantage once again of the prox-heterozygous mice that I showed you before. The lymphatics are defective. They are not normally patterned. They don't transport well. And now to do the opposite. What happened in the resolution of cardiac injury if you have less lymphatic and they don't function well? So what we decided to do, to take these prox-heterozygous animals and just perform myocardial infarction and see how do they repair. And two things were very obvious, very evident. Basically that you can see it, this is sections at the level of the heart, and was very evident. You can see it in red, labeling of the lymphatics in control. You see the number of lymphatics that expand into the injured area. However, you can see that the number of lymphatics is much more reduced in the prox-heterozygous animals. So again, there is a lack of lymphatics. So that impact lymphatic function in these animals, and indeed they do, because when we measure by echocardiography lymphatic function in these animals, the lymphatic function was really compromised in these prox-heterozygous mice 3 weeks after infarction. So this provided additional data now supporting the idea that lymphatics are very important for the resolution or improving cardiac repair after MI. How do they do it? Still is the one of the $1 million questions, because it's most likely it's not just only by providing removing dying cells and providing immune cells to the injured tissue. And here is another evidence, what I just was telling you. You can see that there is increased collagen deposition in the prox-heterozygous animals. You can see it in purple here. Anyhow, that's what I just was telling you. So what is the role of lymphatics then after heart attack? It's just basically by reducing the inflammatory response, or could be lymphatics, increasing lymphatic numbers, and new therapeutic approach. And I told you at the beginning, there are many labs that are following that approach, but in larger animal models by increasing the number of lymphatics using pro-lymphangiogenetic factors, particularly with adenoviral particles, in this case, particularly VEGF-C. So we have generated an animal model that we plan to use in the future, which is basically the same. It's a VEGF-C gain of function mouse model, which in this case, just to show you that works very nicely, this is a control. In brown, you can see the immuno for a lymphatic marker. You can see normal lymphatics in the heart. You can see the lymphatics have clearly expanded in these animals and now cover mostly the entire heart. So the idea is in the future to do again myocardial infarction in these animals and evaluate how much better they respond by having more lymphatics versus a control animal. And the final piece of data I want to tell you about the heart is just is currently in press, but is again a totally unexpected piece of data and another functional role of lymphatics that has to do with cardiac growth. So basically, we demonstrate that lymphatics are also very important in development for the normal growth of the heart, but also for cardiac repair. And but what we were able to identify that is how this cardiac repair is fulfilled by the lymphatics, not only by the inflammatory response, as I was telling, but basically by reducing the cell death of cardiomyocytes in the injured heart. So this is, once again, the more or less the summary of what I wanted to tell you until here about all this novel functional role of lymphatics. And but now what I want to spend the rest of my talk is, okay, that's great, but so could then, you can imagine that particularly in patients that has certain form of lymphatic diseases, so in which we know that the lymphatics do not function as they should, and this could be localized or more global, you can think that there may be a number of associated comorbidities, consequence of that malfunction of lymphatics. And I told you a variety of novel lymphatic functions in health and disease. And it will not be wild to speculate that, similar to what I show you in our prox mice, that they become obese. It could be something similar, that so that a number of these comorbidities are also linked to patients with lymphatic defects like obesity, cardiovascular disease, visual issues, neurological issues, a variety of those. But remember, I told you at the beginning that in our obese prox mice, they are asymptomatics only when we do a deep, in-depth lymphatic analysis, we realize they have lymphatic defects. Otherwise, they look normal. So then one possibility is also to speculate that, similar to our mice, in the human population, there is a number of people that are asymptomatic. They don't have lymphedema, but it could be possible if you inject a dye, like in our mice, they will have underlying defective lymphatics. And if that's the case, you can also speculate that they may have also associated comorbidities, meaning that not all cases of Crohn's disease, neurological diseases, cardiovascular diseases are consequence of the usual suspects, but instead could be partially because of an underlying lymphatic defect. And if that's the case, it may result in better diagnostic and better treatment. The problem is how you address that, how you demonstrate this. Well, it's not easy, but we think we started on maybe in the right track a while ago. And I think the first thing for this is to try to identify a way to, or to identify biomarkers that may allow us to identify individuals with symptomatic or asymptomatic lymphatic malformations. The problem is, don't forget, if you want to analyze lymphatics, all the assays we have so far are in general quite invasive. Mostly you need to inject a dye, do a lymphangiography, and that's not what you want to do, particularly with the asymptomatic potential candidates. So the idea will be to identify an easy accessible biomarker that could suggest that in those individuals, there is an underlying lymphatic defect. And then once you identify that group, then you can go for a more in-depth confirmation that indeed lymphatics are defective. So what we decided was to try to do this, starting by our mouse models, and see if we can identify some type of easy accessible biomarker. And to do that, we decided to use exosomes in blood serum. First of all, it's easy to collect blood serum, so that was not an issue. And we decided to use exosomes. Those are not familiar. It's basically, these are extracellular vesicles that basically trap, or they have as cargo almost everything: protein, lipids, RNA, DNA, microRNAs, etc. and they normally enrich on these particles. We thought that maybe that could be a way that if in those with lymphatic alterations, maybe there is a differential expression of certain biomarkers in those exosomes. So we decided to start this approach long time ago in mice. And to do that, we compare exosomes in wild-type mice, control mice, PROX heterozygous mice, the one that become obese, young mice prior to the obesity, and old mice after they become obese. And as another control, we use the leptin receptor mutant mice, which are extremely obese mice. But we know that in those animals, the lymphatics are normal. So basically what we do, we collect plasma, then we send it to our collaborator, Hector Peinado in Spain, that they purify exosomes, they do mass spec and proteomics, and eventually by mass spectrometry, they identify the protein cargo in these exosomes. All this is published. I'm not going to go in much detail, but bottom line, basically by doing all this bioinformatic analysis and comparing between the controls, the obese mice and the PROX heterozygous, we identify a number of genes that were uniquely, a number of proteins that were uniquely differential for the PROX fat mice. And again, all this is published, but I just want to highlight among one of those that we find upregulated was platelet factor 4 that you can see here highlighted in yellow. And at that time, it was one among many candidates, and we validated by ELISA. This was validated by many different ways. But okay, this still is in mice. What happens, does it happen in humans? And then we started to collaborate with Stan Roxon that runs a very large lymphatic clinic at Stanford. And he was kind enough to collect serum from a variety of patients with a variety of lymphatic disorders and also normal patients. And we have patients with lymphedema, lymphovascular disease, and also, interestingly, with lipedema. Lipedema is one of those disorders for which there is no clear definition of what is the cause of the disease. Some people think they also have accumulation of adipose tissue in the legs or in the visceral area, but there is no easy way to diagnose lipedema, to be honest. So we decided to collect those, and as well as lean and obese individuals, and do the same type of analysis, exosome purification, etc. And this was a very, let me be clear, was still a very small group of people. We didn't have the resources to do an extensive number, but it was significant enough to really give us a hint if we were in the right track. And as you can see here, this is the list of increased proteins in patients with secondary lymphedema, compared with healthy individuals. And once again, similar to the mouse, we found that platelet factor 4 was at the top of that list. Interestingly and surprisingly, we found that PF4 was also increased in patients with lipedema. And that was very interesting because this was one of the first evidence to suggest that lipedema may have an underlying lymphatic defect as well. So what is platelet factor 4? Basically, it's a small cytokine that basically is released from activated platelets during platelet aggregation and promotes blood coagulation. But what is it doing, or why is it increasing in these cases, we still don't know. But bottom line, we finally perform ELISA to really validate our results in all the different samples that we have. And this was extremely nice because it correlated very well, as you can see on the graphics here, among controls, secondary lymphedema, and all lipedema, that PF4 was clearly elevated in all cases. It was only one outlier in controls that you can see here that was also quite elevated. And this was very nice, basically validation of our results, because going back later on to the clinical history of this patient, we found that this patient has all the features of irritable bowel disease that I told you before may have a component of lymphatic defects. And bottom line from this work, I just show you by combining mouse and human samples, we were able to identify now that PF4 is elevated in patients with a variety of lymphatic defects, including lymphedema and lymphovascular disease, but also lipedema. So what we propose from this work is that PF4 is a reliable biomarker, the first biomarker to identify lymphatic defect, in this case, symptomatic. But also we were able to conclude that most likely lipedema has also an underlying lymphatic defect. And very interesting, based on what I was telling you about the outlier, it has been shown also that platelet factor 4 is also a reliable biomarker for patients with irritable bowel disease. One could think now that maybe the reason why it's increased PF4 in these patients is because the underlying lymphatic defect, but that will need to be proven. Finally, in the last two slides, so what I just showed you before during my talk was two things, that there are a variety of novel functional role of lymphatics. And I just show you also that we identify a biomarker for lymphatic disorders. So now what we would like is to combine these two things in a much larger sampling number of symptomatic and asymptomatic individuals. And what we would like to do is a couple of things. First of all, using a much larger sample to really validate PF4 as a reliable biomarker of lymphatic diseases in patients with lymphatic defects, but also by increasing the number of normal individuals without obvious symptomatic lymphatic disorders to identify individuals whose levels of PF4 may be elevated, suggesting that they have an underlying lymphatic condition, and eventually in those patients, go back and do a more in-depth lymphatic analysis to corroborate that they have lymphatic defects in those patients. And eventually to see in that large number, to see if we can associate these lymphatic defects with a variety of associated comorbidities that may have to do with all the conditions that I just was telling you before, from cardiovascular disease, neurological disorders, visual problems, irritable bowel disease, and obesity. I think that in-depth analysis that we hope to do together with Stan Roxon and Hector Peinado may allow us to really determine that in a number of individuals with and without symptomatic or with symptomatic or asymptomatic lymphatic conditions, there may be a variety of conditions that are associated to those defects, and that the main cause is, or one of the contributing factors is an underlying lymphatic malfunction. And once again, that finding will allow to better diagnose and better treat those patients. And that's where we are now, to try to eventually collect more samples and more individuals and generate a very detailed clinical history about these patients. And this is more or less from Stan Roxon, a kind of list among the patients we used so far that were published. You can see that in all these different patients, you have a number of associated comorbidities in those patients, that from cancer, heart conditions, intestinal conditions, etc., etc., etc. And this is the type of database that we would like to create with this in this new study. That hopefully will allow us to better understand all these associated comorbidities. Finally, really to thank a lot of people in my lab and collaborators that have former members and current members that help us move all this field forward, and obviously the funding agencies for this. And now we will go to your questions. Thank you very much. So let me see how to do this. I can see that there are on the questions, there are a number of questions which I will start reading. I may be able to respond some. So let's start with this. John Small Hopkins. They have 2 kids diagnosed for lymphedema 9 years ago, of consequence of FOXC2 mutations. But the symptoms are different. Vascular insufficiency being most prominent. What other documented symptoms and other families or subjects? That's the question. Are the other symptoms? To be honest to John, I'm a basic scientist. I'm not a clinician. So to be honest with you, I really don't know that. But it doesn't surprise me that you have a variety of the symptoms do not match 100% among the 2 kids, because there are many associated situations that may contribute to make one of the phenotypes more obvious than others. But for that, I would suggest just to talk with people like Stan Roxon at Stanford that will be able to give you a much better explanation about it than myself. No. So the same person, Joanne, is asking that they are in Canada. The question is whether there are ways to screen for lymphatic defects. And this is what I was telling you before. Right now, mostly, if they are symptomatic, is relatively obvious. But those which are not symptomatic, still the only ways to by injection of dyes and do lymphangiography. That's why our biomarker could be a powerful tool to eventually at least give a hint. Yes, there may be a lymphatic condition in these patients, and then it may be worth it to do a more in-depth type of analysis. Ron Clark, when you refer to fluorescence in these images, is the RCG, this is called bio-DP, and basically you mix it with the food in the mice, you feed them, you wait an hour, you take the intestines and you look at the fluorescence. Joy Thorne, again, I think these are, unfortunately, I'm sorry I'm not able to answer some of these questions that are more clinical, because it's not my field and I don't want to say something that is totally incorrect. So I will, but he say that basically he has stage 3 lymphedema and also has cardiac disease. Could my lymphedema contribute to all of these? Well, again, it could, based on what I just was telling you, that we know that lymphatic malfunction contribute to many of these things, like, for example, cardiovascular disease. Whether it is a possibility, you will need to talk with a clinician to see your more detailed clinical history. When did you develop lymphedema? When did you develop those conditions, to try to better understand that? But again, all what I was trying to say today is that there is reasonable arguments to believe that in patients with lymphedema or a variety of lymphatic malfunction, they may have associated conditions that are consequence of this defective lymphatic function. Isabel Quer, PROX heterozygous mice exhibit defective and reduced lymphatic capillary network and are prone to obesity. I don't know what's the question, sorry. Is there any PROX? Yes, there's a very good question. So the question is there any PROX variant in humans? Well, so far, no. And reason why my belief on that is that PROX is expressed in a variety of organs and tissues and is critical for the function of those. My sense is mutations in PROX will probably be early lethal. That's why you don't see it. However, a group in Poland described a few years ago a number of SNPs, that means genetic variants in PROX, in 1,000 Polish individuals, that many of those, what they observe is increased visceral tissue, increased adipose tissue in the visceral area of these individuals. Whether that has to do with PROX or not directly needs to be proven. Christine Handler, are you saying the lymphatic system can repair itself after a heart attack? Do you have evidence? No, I'm not saying that. I'm sorry if that's what you understood. Basically, the evidence so far, what they said is that after heart attack, there is increased number of lymphatic. There is lymphangiogenesis. Basically, lymphatics grow and invade the infarcted area and to provide and facilitate the repair. Are there any solutions for patients with lymphedema? Again, all these clinical questions, I think you need to go to a clinician. But for lymphedema, at least I think hopefully we further validate our data. At least now we have a tool, which is this biomarker, to really confirm that you have lymphedema. Because from what I learned from my colleagues in the clinical field, it's not always easy to diagnose lymphedema. So having a biomarker would be a valuable tool. Evan Phillips, do cardiomyocytes in PROX heads have a phenotype? No, cardiomyocytes in PROX heads and even in PROX nodes are normal. And that's an important question because PROX is expressed in cardiomyocytes. So no, cardiomyocytes are perfectly normal in those animals. So Vicky Lydell, so good conditions like rheumatoid arthritis since age 3 could have caused the damage to the lymphatic system. Well, again, same explanation I gave to others. For clinical questions, I will not push it myself. I feel not an expert in this area. But yeah, once again, all these inflammatory conditions like rheumatoid arthritis could really have been linked to the malfunction of lymphatics. Sharon Robertson, is there a known relationship between celiac disease, which causes blunting of the intestine villi, and lymphedema? Again, I think there is a lot of people working on particularly in all these intestinal conditions that may have a correlation with lymphatic defects. Kathleen Francis, did any asymptomatic human show increased PF4? So once again, this is what I said before. The number of samples we did so far is relatively low. However, on the controls individual, the only one that showed increased PF4 was a patient that turned out to have irritable bowel disease. So that's why now we want to expand and basically collect over 1,000 samples of symptomatic and asymptomatic individuals, lean and obese among those, and hopefully identify, particularly among the asymptomatic ones, those that have higher levels of PF4. That will be the definitive proof that A, PF4 is a reliable marker even in asymptomatic conditions of lymphatic defects, and maybe B, find that in those asymptomatic lymphatic defects may be responsible for some associated comorbidities. John Small Hopkins, these mice are my children. All my family are not obese and are in great shape. My son told last week at 13 that pain is visually looking insufficient in lower legs and feet and would soon need compression stocking. How would I know if it's sufficient in the vein systems that we can see? Again, you will need to talk with the clinician, but there are ways that they can eventually visualize deep veins. But again, I will talk with a clinician about that. Harish Paletti, does PF4 have a direct effect on lymphatic structure and on function? What is the possible cause of the increase in PF4 and lymphatic dysfunction? Wonderful question that we will love to know. One can speculate, just speculate. If you think that at least we know that in these conditions, in our animals, for example, you have holes. The lymphatic have gaps from which the leakage occurs. One can speculate that basically what this PF4 is doing is somehow bringing, we know, just let me go back for a second. We know during early development of the lymphatics form from the cardinal veins in the embryonic stages. And during the separation of the lymphatics from the veins, basically platelet aggregates are very important to seal those areas. It could be, this is a speculation, that maybe PF4 is somehow participated in the sealing of those holes by bringing platelets of things like that to those areas. But again, right now, we don't have any evidence for any of that. And this is work that we need to be done in the future. Kelly Bell, is there any timeline where doctors can order PF4 serum test to help doctors differentiate between obesity and lymphatic disorders? Well, this is like with the COVID vaccine. We will hope that it will be here already, but you need to go through a variety of tests first. In our case, the first thing is to really increase the number of individuals, of subjects, such as we have a really significant result that we can confidently say, yes, PF4 is an extremely reliable biomarker. And not only that, but one thing we would like to see is there is a correlation between the increased levels of PF4 and the severity of the disease, for example. Right now, we don't know exactly that because we don't have sufficient numbers. Right now, the major task is to collect enough clinical data and enough samples from symptomatic and asymptomatic individuals to validate PF4, but maybe even, that's the other goal of our study, to identify additional biomarkers that, for example, will identify those patients that have lymphatic defects and obesity, or a different biomarker that identify patients with lymphatic defects and cardiovascular disease, et cetera, et cetera. And that's the goal of our study in the future. But right now, no, we don't have a test kit ready for that. Another question, is there a potential connection between lymphedema as a result of mastectomy and heart disease a few years later? Again, I don't know, and there was the clinical data, how many reports are on that, but again, based on what I just told you, it could be. But in this case, if I understand correctly what you're saying, this is localized lymphedema consequence of mastectomy. So that will be more difficult to do that correlation. What my instinct will say that these associated comorbidities will be mostly linked in cases in which you have a more generalized lymphatic malfunction than in the case you are referring to. Danielle Stimson, did you talk about PROX1 in humans? Is there any association? Well, I responded to that already, so there is no mutation reported yet in PROX1 in humans. Christine Chandler, in patients with lymphedema, use conservative measures to manage their disease. Is this likely that could have a positive effect on comorbid conditions? Similar answer, you will need to talk to a clinician on that. But right now, all what we are doing is to really gather the data to absolutely confirm what I'm just telling you is this a reality. How do your results compare to genetic research at St. George in the UK? these are not just our results. Let me be clear. All the data I show you in all these comorbid conditions come from a variety of labs around the world. Many of these identification of mutations in genes associated to lymphatic diseases come from labs all over the world, and many have been identified at St. George. And the only thing that my lab has done in particular is this identification of PF4 so far as a biomarker. But this is a global contribution, and we need a contribution from everybody to gather more and more data. See you die. PF4 is a marker for platelets. It's possible that platelet coagulation is activated. That's what I said before. It could be. Needs to be demonstrated. Flavio Sousa-Smith. Have you immune profiled the perilymphatic adipose tissue from the PROX1? We haven't done such a detailed analysis, but what we know that the adipose tissue is really invaded in these animals, at least by immune cells and macrophages. So we know that adipogenesis eventually is an immune triggers an immune response. So it's somehow not surprising, but much more will need to be done in that respect. Lisa Fisher. If we can test for PF4 in someone with a symptomatic lymphatic feature, may have a diagnosis of Parkinson, Crohn's, etc., how do we present this to the physician? And if they agree to do these tests, how we can proceed to treat these patients or maybe improve their diagnosed condition, or is this futuristic? Well, it is futuristic in the sense that it will take us a while to gather more data, but at least maybe I'm being naive, but I think it's promising because I think we are finding more and more data, which is partially what I presented to all of you about the role of lymphatics in all these conditions that I mentioned. And whether it will be PF4 or other biomarkers in the future, that will need to be determined. But I think for a clinician perspective, once they have enhanced some strong data that argues, well, your whatever comorbidity from Crohn's, heart disease, maybe partially also triggered by an underlying lymphatic malfunction. I'm not saying we can cure the lymphatic malfunction yet, but at least you know, or the clinician will know that those conditions are not only triggered by the usual suspects in the cardiovascular field or in the Crohn's disease field, that maybe there is another component, which is the lymphatic, that needs to be considered. And I assume that in the future, all of those pieces of data will allow for a better diagnosis and eventually treatment of those conditions. But again, I'm not a clinician, I'm just telling you my sense of this. And I think the more we learn, the more in the future we will be able to really treat those situations. Matthew Borchiel. Do you think the PF4 is being produced by dysfunctional legs or is a consequence of the underlying environment in disease? Great question. And to be honest, I don't even know if PF4 is produced by the lymphatic. Let me be clear, by the lymphatic endothelial cells. Just to be clear, we isolate the exosomes from blood serum. So what is the source of PF4? We don't know. All what we know is that it's increased, but whether it's produced by the lymphatic endothelial cells, the blood endothelial cells, or whatever other type of cell, we don't know. For us at the moment, it's irrelevant. All what we know that is increased. And that's what matters right now, because what we were looking was for a biomarker. Hanshoffs, what is the mechanism for the leakage of chyle in heterozygous animals? Are junctions protein affected? Yes, we know that there are a variety of junctional proteins affected, downregulated, but exactly the reason we don't know, to be honest. What we know is that you find huge gaps in many areas of the lymphatic vasculature in these animals, through which, again, whatever content they have, they leak out. It will be interesting maybe at some point to better understand what is the reason for that. But one possibility we speculated in the lab many times in the past was that not only the junctional proteins may be affected, but maybe what could happen is that basically in these animals, the vessel is formed by less lymphatic endothelial cells than in a normal condition. Therefore, as a consequence, basically you're really pushing the cells, and therefore the junctions become much a liability, if you like. But again, we haven't confirmed what is the reason for that. Is there anything on the horizon? Schiller Hill, to promote lymphatic regeneration, which fortunately not yet. Again, depends. There is a number of groups. They are trying to use VEGF-C as a lymphoangiogenic factor to really regrowth lymphatics in certain instances and in certain conditions, and that seems to work. Whether this could eventually work in more generalized conditions of lymphedema is still far. But once again, I think we're at a point right now compared to where we were 20 years ago that I would not say no to anything, because I think we learned so much about lymphatics that I think there is reasons to be optimistic in the future. Sarah McCollum, I have lymphedema that presents only in one area, left leg. Is it still possible that the localized effective lymphatic could be related to neurological, cardiovascular? Well, again, that's what I addressed before. To be honest, we do not know that because, again, the number of clinical studies doing all these correlations with comorbidity is not so large. And this is a great thing. It's just unilateral malfunction sufficient to lead to some of those comorbidities. It's hard to say. I will tend to believe maybe not, but I really don't know. We don't even know exactly why sometimes lymphedema is in one leg and not the other. So again, I think there is a lot of things we need to better learn about this. But I think we are at the start of this race, and I think, again, as I said before, I think we should be optimistic about it. But how these associated comorbidities associated with what type of lymphatic disease or how broad the lymphatic disease should be, we don't know. Again, in the larger study, which we will include all these patients, we may have more clinical data to validate this. What we know in patients with lymphedema, they have elevated PF4. It's about correct. Thanks for your answer. I still know you answered my question. Patricia Redondo, what is the role of LE&RN positive cells that were not forming lymph vessels in the heart image? Those are macrophages. LE&RN is also known to stay in a subpopulation of macrophages, and that's what you saw in those images. Patricia Redondo again, do you have any evidence that could suggest how PF4 be altering the lymphatics in any of these conditions? Unfortunately, no. Hena Kumura, how are lymphatics affected in cancer patients who are obese, given obesity being a risk factor for many cancers? Again, there is data that suggests that in obese individuals, lymphatics are somehow impacted. But again, I think this is something that needs to be analyzed in much more detail before reaching conclusions of this type. Catherine Francis, Brock's heterozygous mice exhibit similar GI, cardio, CNS, and other comorbidities as humans with lymphedema. Yes. I don't know if this is a question or a statement, but not necessarily. But what we know so far is that yes, PROX, heterozygous mice exhibit obesity, issues with cardiovascular disease after MI, but whether they have issues, CNS issues, we don't know, and it doesn't seem to be the case. But this is a mouse, so it's hard to know precisely whether that's the case or not. And PROX is a complicated gene to do these studies because half a copy of PROX impacts many cell types as well and many tissues. LE&RN, are you seeking external clinical subject samples? So, well, we are going to start, we are still waiting for funding from different sources to move forward with this. These are not cheap studies. But basically, the major clinic that will collect sample is the one from Stan Roxon in Stanford, and we will have some colleagues in Poland as well that will be collecting the samples from individuals. Kimberly Johnson, are there any clinical trials we can sign to give more info on PF4? Once again, the contact eventually will be Dr. Stanley Roxon, but it's not just thank you very much for being willing to participate. What we need in addition to the blood serum is eventually a very detailed clinical history and characterization of all the features. So that's because that's the two things we need for this story. Evan Phillips, can you talk more about the relationship between cardiac lymphangiogenicis and fibrosis? All what I can say so far that the studies done so far seems to suggest that increased cardiac lymphangiogenicis somehow reduce the fibrotic area after MI, but that's as much as I can say, and probably based on our recent data by reducing the number of dying cardiomyocytes. Hari Shankar, my son has primary lymphedema in his right leg, but also has osteomyelitis. Is there a connection between the two? I will suggest you need to talk. I really don't know. There is some data of lymphatics in bone in certain conditions, but you will need to talk with a clinician on that. Diana Trapani, is there a difference in PF4 numbers between primary and secondary lymphedema? Well, because the numbers are still not large, it's hard to reach a strong conclusion because even between individuals with primary or secondary lymphedema, we see some variability, whether it's because the severity or when they started or the age of the individual. These are all the features that we need to have a large number of samples to be able to reach all these to address all the questions that you guys are asking me, which are very important. But again, these clinical studies to require a large number of people to reach all these important conclusions. At the moment, I don't know. So I don't see more questions here on the questions and answer. Okay, in the chat, sorry, there is a lot of questions in the chat. Let me see.
Dr. Oliver, I think most of the questions in the chat box got transferred over to the Q&A box.
Oh, okay. Then if that's the case, I think we are done. If there is no burning question from anybody, thank you for participating. I hope to have been able to address some of your questions, and I hope that you guys are all excited about the field. I think the field is in a great moment. It's a lot of things going on, and have a good day. Thank you very much.